This is a production of Cornell University.
I'm all mic'd up and ready to go. Thank you very much for the introduction. She made my research sound really incredibly exciting. I'm going to talk about none of that today, of course. I'm just going to talk mostly about the MyCoPortal, but I'm more than happy to talk with anyone afterwards about my research. Thank you again for inviting me today, and let's get started. Let's see. Okay, so before I introduce the MyCoPortal that runs in Symbiota software, I kind of want to give you a little bit of background about how we got from point A to point B, from there to here. Basically, there was 8 or 10 of us mycologists sitting around a bar after a meeting, wondering all this data that we have in our collections, how are we going to get that off of the labels and into a digital format where it's somewhat useful for the general public and for other researchers to use, right? And I guess I'll spoil the story and say, well, fast forward to today, and we're pretty much there. So we've done it. Unlike the some of the botanists that have a long way to go because there's a lot more plant specimens, or the entomologists that have a lot more insects to digitize. Regarding the fungi, pretty much I could say with some confidence about 95, maybe closer to 99% of all the herbarium fungal specimens in Fungaria throughout the US have now been digitized and is available here on the MyCoPortal. So you can imagine, vast amounts of information here on this label data, and we need to get it out there so people can use it and correct it. Sometimes all this label data isn't exactly correct. So we find a lot of things in small, large packets in boxes, and then in these Exicati sets as well, that's pretty much hidden, in some back room or even off campus in some location where it's not all that accessible. Excuse me. And there's various ways to get this information off of the label and into a computer. If anyone's familiar with some of the Global Plants Initiative from years ago, they had a large grant, which is pretty much 100% success rate. You just ask them to go and digitize type specimens. So most of the plant and fungal type specimens are done thanks to GPI, and they use these really large, high-resolution scanners, which is fine, but they're very slow, and you're not going to be able to do many of those an hour. So the digitization projects at NSF have recently funded pretty much rely on similar protocols to use camera systems, and we're looking at a rate of 40 to 50 per hour. So close to 1 per minute, taking pictures of these label data really, really quickly. This is kind of the old-time way of doing it. You got your undergrad there with his headphones on, listening to some sort of music or whatever, and basically just keystroking all of this data in. I know NSF likes to fund projects that are transformative, right? They want something that's really, really the next generation thing. But when all this is said and done, even if you take these pictures and you use OCR, optical character recognition, along with natural language processing, in the end, what we found based on side-to-side studies is good old-fashioned keystroking is really kind of the way to do it. Even though there's a lot of these new and innovative techniques, which are very cool, but sometimes just sitting here doing it like this is the way to go. So some of you may be familiar with some of these different collection management systems, the different softwares. You may have heard of Arctos, which is kind of invented more for museums, and a lot of the vertebrate people use it. KUMU is for those institutions with lots of money, like the Field Museum and New York Botanical Garden. It's quite an expensive type software. Good old FileMaker Pro or Microsoft Access or Excel. There's a Paradox, kind of a government one. And then Specify and Symbiota are two of the larger ones that's kind of out there now that most collections are using, biological collections are using to manage their herbarium collections or whatever entomology collections. So today I'm going to be talking about Symbiota, because that's what the MyCoPortal runs in, is this Symbiota software. So what is Symbiota? It does a lot of things that a lot of the other softwares do, although it does some things really well that others don't. So it's first and foremost a specimen search engine that's out there on online portals. It serves a lot of different floristic data through specimen checklists and surveys. It has built-in identification keys. It has this massive image library. You can produce different types of distribution maps, which I'm going to show later in my talk today. You can produce descriptions. It has a host of other different taxonomic information. You have the ability to enter genetic data and link that data to and from GenBank, which I'm going to show you later in my talk as well. And one of the more powerful things is it's a data aggregator. So we can actually go to GBIF and we can download other collections and upload those into the MyCoPortal. So we actually aggregate everything into one place, which is really one of the nice features of Symbiota, is being able to aggregate all of this data, and you have one place to go and search for it. So Symbiota software, for those techies out there in the audience, it runs on PHP. It has a MySQL database in the background. The developers made it completely open source, so it can really never be licensed and sold down the road. It has to remain open source. It's a modular system where they have different specimen, floristic, and identification modules that you can kind of plug into these different portals. So there is some customization if you so choose. It's quite vast, and a lot of people have bought into Symbiota. So there's 43 portals as of today, serving somewhere around 37 million records in total. And if you read the names of a lot of these, you'll see that they're either regional type portals. So for example, intermountain region of plants there in the herbarium network, or they're more taxon specific type portals. So you can imagine like the macroalgae herbarium, what it contains, LepNet, you know, that's where your butterflies are going to end up going. And of course the mycology collections portal, that's where the fungi go, except for lichens, because the lichenologists wanted to have their own portal. So there's also a lichen portal as well. And there is some talk about combining these down the road, because as we all know, lichens really are fungi, right? But anyway, there's just a few key Symbiota websites if you want to check out more information. There is a home page. All the code is at a GitHub repository, so that's where all the open source code is. Any developers or programmers can come in and change the code as they like, and then those changes are either accepted and put into future versions of the code. There is a single publication that came out several years ago that explains kind of what Symbiota does. We do have a Google group support group, which is quite nice. If anyone has any questions, hey, how do I do this? They just email the group, and then there's always people out there to answer it. I'm co-chair of the Symbiota working group that hosts webinars from time to time. So you can go to the iDigBio website and look at our wiki page here for the Symbiota working group to kind of learn more. We do have different webinar series. All of these have been taped, so you can always go back and listen to these webinars. Most of them are about 30 minutes to 45 minutes long, and we talk about a certain subject on how to use Symbiota. So there's a lot of resources out there. Just for those not familiar with iDigBio, that's kind of the hub from NSF. I'll talk a little bit about that later. That's kind of the realm down at the University of Florida, where Larry Page was the head of that for several years. And that's what sort of organized all of this digitization project in the United States. So we do have a mission statement for the Mycology Collections Portal. The idea is to provide an immense resource for aiding mycological research and education by connecting this information generated from its user community to educators and researchers throughout its network. So that's our primary focus, is really what we're trying to do, is get the data all into one place so researchers can use it, especially grad students. So for example, if you're working on bipolaris and you want to know where all the bipolaris are in herbaria, if you want to go and study those, 5-6 years ago, you had to go to each individual herbarium website. You could do that 100 times and do 100 searches if you really had nothing better to do with your time. Now, thanks to the MyCoPortal, I'm proud to say you can go to one resource and find where all these collections are, just with a single search. And that's really the whole point of aggregating all of this data together. So our homepage for the Mycology Collections Portal, we have this fun little fungus of the day here. If you want to test out your taxonomic skills, you can go and take a look at that. Excuse me. As different images of fungi get loaded up, we have this nice little slideshow that kind of displays different fungi. And then of course all the regular tabs. I'm not going to take you through and show you everything, how to use it in MyCoPortal, but just some of the highlights down here of what we do. It is it is user controlled. We have permissions for each of the different collections in here, and you can be a data editor or an administrator, giving you different permission and rights to go on and use it. The general public, you don't need any user logins. So don't worry about another username and another password. You can just go on and use the public interface. And that's primarily what I'll be showing today, a lot of the public interface for people. So how did this thing all get started? Well, for those not familiar with the NSF program, ADBC, it stands for Advancing Digitization in Biological Collections. And NSF did something a little bit unprecedented. Other than NEON, they typically do not fund large programs for more than 5 years. In this case, they decided to put in $100 million into the pot over a 10-year period. And that's what came about of all these different digitization projects. And they typically fund somewhere between 2 to 4 of these multi-million-dollar grants each year, starting in 2010 was the first round of funding. So the MYCOLOGY collections portal started out with a macrofungi collections consortium led by Barbara Tiers here at the New York Botanical Garden, along with lots of help from Scott Bates, who's up at Purdue Northwest in Indiana. And the first grant to help fund the MyCoPortal was a $3 million grant, which ran from 2011 to 2015. As the name implies, it was pretty much directed at macrofungi. And then we lined up a large group of microfungal experts, and everyone took a step back, except for me. So unfortunately, that's another way of saying I took a step forward. I don't know how many people get that joke, but I was pretty much it. No one else really volunteered to head up this thing. So I decided to go ahead and write the next logical piece to the puzzle was to do the microfungi collections consortium. And basically, I told everyone in the network, look, we did all the macrofungi. Lichens had their own grant, and they digitized the lichens. Let's just clean up everything else that you have left that's a fungus, right? And most of them were microfungi and slime molds. And pretty much anything else in your fungarium, you need to go ahead and digitize and finish it off. And that's pretty much what we ended up doing. As Kathy stated, we had 38 different institutions involved in this. I'm not sure how many of you have ever put 38 PIs, or I should say cats, into a room and then tried to herd them in one direction. I think at the end I counted up, it was just for fun, it was like over 2,000 emails and about 300 different phone calls to get this all together. Because we had 22 budgets, 22 bio sketches, 22 current and pending support, 22 previous NSF. You know, if anyone's wrote an NSF grant lately, you know how many pieces there are. Well, multiply that by 22 with the sub-awards. It was quite a large network, and I will never, ever do it again. but it was fun. It was fun. It was fun while it lasted. It was just a large project. So we got another $3 million, which ran from 2015. This should probably say 2020. We just got another second year no-cost extension grant for another year of funding. We have a little bit of funding left to kind of finish up the project. So $6 million does help, right, to get things accomplished. And that's what we can thank NSF for in order for getting a lot of this stuff digitized. For those of you on Facebook, we do appreciate likes. So if you would please go to the microfungi, I think it's just microfungi community on Facebook. You know, NSF likes numbers and stuff. So if you like us, I can report that next week in my annual report that's going to be due soon. So we do put up, you know, interesting little stories about fungi in general. This was our first network meeting in NSF. Kathy's here in the front row, so you know, proved that she was actually there with us. She wasn't off somewhere else goofing around. But this is our network of all of the different contributors that came. We met in Washington, D.C. for our kickoff whenever we started digitizing, just to make sure we were all on the same page. And that was during one of the NSF iDig Bio summits that we have every year to get all of the groups together to talk about digitization tricks and techniques. So like I said, we had 38 participants. Of course, it's US taxpayer dollars, so we could only fund people within the United States, actually give them money to digitize their collections. This was some stats from last year, where we had 171 people in the network, so quite a large network of people. 27 PIs, of course, that don't do a whole lot of work. It's all these other people that we rely on for the real nitty-gritty, right? So 22 project managers, 28 other paid staff, 3 grad students, here's kind of the important take-home message here is these 80 undergrad students, which really gets underrepresented. We really don't point that out enough or focus on that enough, is the fact that these undergrad students are doing a bulk of the work for us, but in return, our taxpayer dollars are also going back to the undergrads. So some of them would stay around and work 2 or 3 years on this project, and it really was a significant financial incentive for these students, help pay off for their loans or whatever other expenses they had. So I think that kind of goes without saying sometimes how much of the money was actually returned to undergrad students helping us do the digitization work on this. And so I just wanted to point that out. So right now, with the entire network, with the data aggregation and other people coming on, maybe once every other month, we get a request, hey, can I be part of the MyCoPortal? And we add them on to the collections. So we've now kind of hit the centennial milestone here. We have 100 institutions within the MyCoPortal right now, primarily throughout North America, although we are gaining more in Central and South America, and we're expanding over into Europe and Asia as well as we add these on. So this is a list. I don't expect you to read all of these, but this is a list with all their logos of all the different 100 institutions that we have throughout the MyCoPortal. We have them divided here into those that have specimen vouchers, but you can also serve observational data as well. So we brought in Mushroom Observer and iNaturalist research grade observations as well. And observation is nothing more than, think of it as a bird count, right? They've seen the fungus, they've taken some pictures, but they haven't accessioned it into a fungarium somewhere and actually housed it somewhere. So you can't go back and get the specimen voucher. All of these others, all the data is backed up by actual specimen vouchers in an institution. So the really nice thing that I'm most proud about is that 62 of these 100 collections are live. And what do we mean by that? They're using the MyCoPortal as their primary database at their home institution, okay? So they're not using Specify or KEME or FileMaker Pro or Excel and then uploading the data. That's what these snapshots are doing, actually. They're using a different primary database, and then we just grab it through an IPT. So for example, the New Zealand, not only New Zealand culture collection, but the New Zealand PDD herbarium, both of them periodically every few months, we just go out and click a couple buttons. Because everything's in a standardized Darwin Core format, we can just grab their data and it just sucks it right in, the most updated data right into the MyCoPortal. A lot of these are smaller institutions that really can't afford some sort of primary database on their own. So I'm pretty happy to be able to provide the MyCoPortal as a service to them. Others are quite large. So University of Tennessee, University of Arizona, some of these really large institutions they're using the MyCoPortal as their primary data. So when people ask me about sustainability, I kind of jokingly say we're like the banks. We're too big to fail now, right? We pretty much have to keep going because 62 of these institutions throughout the US and Canada are using the MyCoPortal as their primary database. So that's what they upload to, not upload, that's where they enter their data every day. So as I said, it's a large network. We had to have a large management team to run all of this. So we had various data curators. We had Teresita until Kathy came along and headhunted her and brought her here to Cornell. So, you know, you train up good people and then they leave. That's just the way life works sometimes. But we still have a biological informatician that helps with a lot of the background, you know, data networking and a lot of the background tools that I don't understand the computerization of it. Scott's still hanging on as a project consultant, helping us out of various different undergraduate students. And a retired emeritus professor, Lee Crane, who's really a Exicati and nomenclatural expert. And as we speak right now, he is sitting in the plant pathology herbarium working on your very own Exicati right now. So we drove out here from Illinois and I brought him along because he wanted to visit Cornell. So he's helping you guys right now. I wouldn't let him come to my talk. I told him he had to keep working out there. So, but he's working on your Exicati in the plant pathology herbarium and going over a lot of those. So, so it's great to have him along as an expert to help with a lot of various aspects of this project. We have a lot of resources in English, and thanks to Teresita, also in Spanish as well. So all of these have been translated. If anyone wants to go on and read sort of these very quick how-to guides on how to upload data, how to deal with Exicati, how to make a checklist, and also linking data between Symbiota and GenBank, things like this. They're very simple. There's not a lot of words. We wanted to make it very pictorial. You know, it's kind of like when you get that bicycle for your kid for Christmas and you have that quick how-to guide. You don't want to read the big manual on everything. You just want to read and get up and running. So a lot of these are only 1 or 2 pages with a lot of arrows saying go here, go there, and screenshots pointing it out with not a lot of verbiage in there to read. So it's quite easy to get up and running quite quick on learning how to use the software and entering the data. So at last count, we're also very proud to say we've got 4.5 million records in the MyCoPortal right now. Of these, over 4 million are tied to specimen vouchers, and the other 500,000 are the observations that I had talked about earlier. We're not bad with about 50% georeference so far. So this allows us to ask a whole lot of questions, right? Once you get geocoordinates, you can put a dot on a map, but you can ask a lot of questions about invasive species movement and distribution maps, where they occur, things like this. Of these 4.5 million, I also want to include or just say that we have over 115,000 type specimens representing 45,000 taxa that are actually type taxa in there. And at last count, this only started in 2012, the MyCoPortal, but we now have 46 peer-reviewed citations. So we have these actual, not just something mentioned on Facebook, but actually peer-reviewed journal publications that have went to the MyCoPortal, used our data, and cited us. So that's really exciting. So again, lots of collections. You can click on any one of these collections and pull up all of the data on it, right? So a lot of this comes from Index Herbariorum, if the collection's in there. So this is Cornell, or CUP is your acronym if you didn't know that. It tells you who the contact is. You can go to the homepage to learn more about that herbarium, what the collection type is, and then a little bit about the collection statistics, how many that's in there and how many is georeferenced, et cetera, things like that. So we've been helping Cornell out also with some of the georeferencing, and we're sitting on about 46,000 more records that have been batch georeferenced that hopefully we'll add in the next week or two to Cornell. So as we finish up our collections, we help other collections get georeferenced and get their data up and online. So I'm just going to show you kind of the public search interface here with some examples. There's a lot of specimen records, or some specimen records out there called Cornell Woods. So we look at we type that in locality, and you kind of just hit search over here and find out that there's 48 records. And obviously a lot of them are in Cornell University, right? Those specimens have been deposited there. But maybe a little bit surprising is there's a bunch of other institutions that have your specimens in their institutions, right? Including New York Botanical Garden, Cortland's not a big surprise, BPI, but Arizona, Berkeley, Washington State, Michigan, Georgia. These other people have come to Cornell Woods, whatever that means, and they've collected specimens and put them accessioned them into their herbaria. What we have is a couple different taxonomic thesauri in the background. So one of the ideas behind the mycology collections portals, we didn't want to be GenBank. We don't want to duplicate what they do. We don't also want to be Index Fungorum or MyCoBank. We don't want to be a taxonomic thesaurus constantly changing names. So what we do is just bring in these taxonomic thesauri, and you can switch between Index Fungorum or MyCoBank because they're taxonomic thesauri. They give you slightly different results, right? Slightly different names, and you can choose your taxonomic thesaurus of choice. But you can select MyCoBank, which is the default one, and you come up with 48 different taxa, and you kind of get a species list of all of those that occur at Cornell Woods just by clicking the species list tab over there. And now I'm going to digress a little bit and just talk a little bit about the Spatial Module. This was developed over the last couple of years. It's really quite exciting because it allows you to do a lot of different things. This is one of those exciting modules we're happy to offer. It has a whole range of visualization options with different occurrence point maps, right? So because these things are georeferenced, you're able to put dots on maps. So you can learn about phenology, and there's been several papers already published on the timing of fruiting bodies because of climate change, right? Things are starting to warm up faster, and some of these deadly amanitas and these other edible morels are starting to fruit sooner because of climate change. Well, we can look at a lot of that because all the data is in one place. You can look and see how invasive species have changed through a data slider. So every 10 years, if you want to see the movement and track the movement of those species, you can do that. And then there's a host of other things like range maps, suitability maps, climate chains. There's these heat maps down here. And it's really useful for outlier identification, right? If you got something off in the ocean and it says Pennsylvania, well, you need to go back and look at that data and correct the geocoordinates. So for more techie people, we offer vector and raster layers both, and you can upload your own vector data as well in some of these different formats and do more things. So the spatial module is quite powerful and does a lot of interesting things in the MyCoPortal. So here's the map of Cornell Woods, right? When you geolocate, and whatever Cornell Woods means, you have these different points on the map. So you can kind of go and look and see where these different things have been collected. Obviously, I said there was 48 different collections, but not all of them have been georeferenced. So there's really not 48 things here on the map because we don't have geocoordinates for everything. But there's other things you can do with the spatial module that might pertain more to the crowd I'm talking to, the plant pathologists. So I just Googled Plasmopara, and I've color-coded each of these different species based on what the species are. So you can color-code them throughout the world and distributions based on species. Here's an example from Phytophthora. Most of you can probably guess which one is Phytophthora infestans. It's the one here in bright pink, right? That seems to be the most prevalent and occur throughout the world. Although ironically, we don't have anything from Ireland up here, right? Or over in here. I don't think there's actually one from Ireland, even though it's Phytophthora infestans. So we'll work on that. We'll try to get some specimens of Phytophthora in there as well. I also heard that Cornell came into a little bit of money as well recently from the USDA grape lab. So I thought I would use this as a nice example. The black rot of grapes, just Googling that taxonomic term, we can kind of see the distribution of that pathogen that occurs on grapes throughout the US. These little triangles are the observations. All the circles are backed up by a specimen voucher, right? So these were just observations, but we have a large range throughout the central and eastern part of the US of this pathogen, according to the MyCoPortal. The nice thing too is that we actually added a host field as well. So if anyone's familiar with Darwin Core, it takes about 6 years to introduce a new field to Darwin Core and then to get it blessed by all the powers that be. The plant pathologist said, we're not gonna wait that long, just we want a host field. So I said, okay, all right, you're the boss, that's fine. So we put a host field in here as well. So host is not really a recognized field. For whatever strange reason in Darwin Core terminology, it's not. It seems quite important to me as well as many other plant pathologists. So we added this host field, and now you can search by host. So just the common grape here pulls up 574 different records from a variety of different institutions. This is just anything and everything that occurs on grape. So again, here's the host field. And throughout the world, these are all the different taxa that occur on. So there's over 500 records, but when you select a taxonomic thesaurus, there's actually 154 taxa, right, or species that are associated with grape in some way or shape or form, right, as host. You can also map these things. There's not obviously a lot of dots on here, not a lot of things have been georeferenced. I showed you a Google map before. You can also map these things in Google Earth by downloading, I think it's a KML file, and then just putting it in Google Earth, and you get these little pins that stick up here. Another search that I did just for fun was the host ZMA. So looking at corn, in addition to the genus Fusarium, we find there's 85 records in there. With the MyCoBank taxonomic thesaurus, we have about 14 different species of Fusarium, right, including Gibberella as well. So as we get these anamorph, teleomorph names kind of linked up in the taxonomic thesaurus, it'll give those names and all the synonymies in there as well. So we don't have to worry about that. A lot of people ask, well, what name do I put down? I have this name on the label. I said, it doesn't matter. Just write the valid, as long as it's a valid species name, just put it into the database, because the taxonomic thesaurus will figure out all the synonymies in the background. We don't have to do that. So that was kind of a nice feature. And again, you can go and search it using the Google Earth with different kind of features as well. So it depends on what kind of map you want to produce. You have a variety of options to do that. So I just wanted to briefly kind of point out that there are a bunch of different checklists on here as well. we've kind of divided up into fungi across the world, historic collections, fungi in North America. There's macrofungi and different, there's a microfungi. And these are kind of divided up based on geography. So, you know, countries or different groups, local and regions, and different people can use those to create their checklists. And then there's some organization to these different checklists. So for example, if we wanted to look at the macrofungi of North America based on various regions, mostly states, we see, you know, New York doesn't really have a checklist right now. No one's went on and produced it, but the closest thing was Maine. So I kind of pulled Maine up and just show you that, you know, a quick checklist of Maine for macrofungi shows that there's somewhere around 140 families, 545 genera, and around 1,600 different species that occur throughout Maine. So you can do things like this. Now that the data is in there, you can actually find out where they occur for your favorite region. And you can imagine, you know, school teachers, if they wanted to, going out to a central park, a local park, and pulling up a checklist to see what they might find. Maybe not so exciting with fungi, but they can do this again with birds or butterflies or any of the other Symbiota portals. They all work the same because they're all using the same code. So, and then I'll just briefly mention Exicati. We have a very large list where we put in all the different titles of all these different Exicati. There's 491 Exicati titles in there now. Most of them are populated. Some institutions are the only ones in the world that have a single copy of these Exicati. Sometimes, like North American fungi, are quite common, and you know there might be 20 copies throughout. But we were really looking for those rare ones so we could get all the fungi in Exicati put into the MyCoPortal as well, and we're still working on some of that. If you search Mycotheca brasiliensis, for example, that Exicati, and we do have some duplicates. So Brown University and LSU both contain that Exicati. They've digitized it. There's a picture up there. If we just go to look at the label picture, you know, at Brown University, this is kind of what we're seeing right now is that label data along with the barcode. Okay, and that's pretty much how the digitization project worked. What we would do is take a picture. We would make kind of a skeletal record up here with maybe just the name on here. And then we'd have the undergrads come through. This is how we got the data in, and they would transcribe the information on this into all of these different fields, whatever the pertinent fields were. So we have this Exicati title and then the number 10 right here. And that's how we got the data in, basically by taking a lot of pictures and doing a lot of transcription. Once you have locality data in, then you can batch to your reference, and then that's where you get your latitude and longitude filled out here. So we kind of have these different stages, okay? All right, so I'm just going to switch briefly about another module that we developed the last year of this project, something that I'm pretty excited about, I think is pretty cool. It's called the MyCoPortal ITS sequence submission tool. Actually, we should just say that's a ribosomal one. You can submit ITS data, large subunit data, and small subunit data. It's very similar. If anyone's went on and used GenBank's online portal recently to submit sequence data, you'll know that you can only submit ribosomal data through the GenBank portal. If you have protein coding genes, you still got to go back and use Bankit or Sequin or something like that. But basically, we started thinking, we have all of this data in the MyCoPortal. Why not just submit all we need is a sequence, right? That's really the last piece that we're missing, because what this tool does is it grabs your user profile information, because you'd need to be a user and have a login to submit sequences through a collection. We already have all of that data. We have all of the specimen metadata already, right? Because that's what we've been working towards, typing in that specimen metadata. All you really need to do is paste in your sequence and then send that to GenBank. It seems silly to type all that information in a second time. And this was one of the primary instances why we decided to go and do this, is I did some searches in GenBank last year for what they call source modifiers. That's basically the specimen, another word for specimen metadata, but GenBank refers to it as source modifiers. They're seldom populated in GenBank, right? So we can look at all these different primary source modifiers for all these different groups. And it doesn't really matter if you're a mycologist or an entomologist or a botanist or a zoologist. We all do a very poor job at populating our specimen metadata that's tied to sequences, right? So picking on the entomologist and picking on the botanist here a little bit, I know Kevin Nixon's in the background and will really appreciate this. You guys are really bad at submitting specimen, even the general data like specimen voucher data. Only at 1/3 of the time can you go and figure out where the specimen, what the specimen voucher is, it's tied to that sequence, right? You have no idea. Sure, Kevin. You don't even have vouchers. You just sequence it and throw it away? Okay. Yes, that's even more of a problem. Exactly. You can't even go back and recreate that again if you wanted to, because the specimen voucher doesn't, no, of course not yet. Of course. So, you know, you can kind of look at all of this stuff. Some people are better than others. The entomologists seem to like to populate country better than some of the zoologists do. Isolation source or even host data, but a lot of these are near zero, right? On who collected it, collected by, the collection date, who identified it. Sometimes that's really important, but we just don't really have that data. Longitude data and altitude. So the reason, the emphasis behind creating this tool was all of this data right here already exists in the MyCoPortal. Why not just capture that data when you submit your sequence? And then all your source modifiers in GenBank will be populated. So I'll just very quickly kind of show you what this tool looks like. It's not a training session, but it grabs your username up here, the record ID. So yes, it is a little cumbersome because you can't batch submit sequences. You have to do it one at a time. But it grabs that record ID. So now it knows where all that specimen metadata is. You basically just paste in your FASTA formatted sequence. You pick whichever one it is. Like I said, it's ITS, LSU, or SSU. Some people may do just a part of ITS, and you can choose that too. This is very analogous to what's in the GenBank submission portal. Almost everyone's doing Sanger deoxy sequencing, but if you're not, if you're doing some Illumina or something else, you can select that. You can type in your title of the publication if it's published, and you can add your authors right here. So that doesn't take too long. Here I added a couple, or you can delete them. You can either embargo it for a year or not embargo it. It just depends on whether you want your data released immediately or not. And then you basically just click go, and what it does is end up building these GenBank formatted data sets. I'm happy to say, other than Genius, Symbiota is the only other people in the world that can submit sequences to GenBank using a third-party resource, right? And it took a lot of meetings back and forth with the GenBank indexers, and they were saying, no, do it this way, do it that way. So I did it the way they liked, and now they accept this data. And what you basically have is a sequin file. You download and just email it to GenBank, and you have this pretty cool file called a GBIF file. And this is kind of what you see is what you get. So this is what your GenBank submission is going to end up looking like after you submit it and they publish it. So you have your publication authors here that you just put in. You have the submission author, whoever submitted that. The institution, all this stuff is directly pulled from the MyCoPortal. I'll just briefly go down here and just say, here's the specimen data, these source modifiers that I just talked about. It's all populated right here automatically. And the cool thing is we also developed a structured comment, what GenBank regards as this kind of cool little extra thing for developers and programmers, but also for collection managers as well, because what it does is give you the record ID or a GUID. It also gives you a GUID if we had one, which is a globally unique identification number, right? So you know exactly, it only has one number in the world that link back to that specimen. It tells you the source where it come from. If you're using a different portal, that would be populated. The institute, the catalog number, which is repeated, and some other information about the accession number. But what this allows programmers and developers to do eventually, if this gets populated enough, is they can just go on and write a little MySQL search statement, and they can grab all your data out of GenBank much easier if you have this structured comment in there. So it's just helping to make the world a little bit smaller, hopefully. As far as genetic data goes, we have these linked resources. So the identifier, where it came from, that's the GenBank accession number, what the gene is, and then a URL link. If you click on that, it takes you to GenBank. We're also linking back as well. So we're using this link out system to go back to the MYCOLOGY collection portal. And if you click on that for the ones that are linked, it takes you back to the public interface here that shows all the details about that collection. This one's at University of California, Berkeley. So it's kind of a neat resource to link to and from specimens. So what do we do with all of this data? And Kathy alluded to this a little bit earlier. Is a project my colleague and I did, along with all the other, tried to give as much recognition to the network as we could. All the other people that helped put data into this is we created the first ever, what we call a proto checklist. But because by no means is this a final word, that this is a checklist of North American non-lichenized fungi. There are a lot of mistakes in it, but we needed something out there, right? If you don't have anything out there, you can't correct it. You can't move, science doesn't move on. So we went ahead and put that out there. Some of the highlights of this paper was we looked at over 2.2 million fungal records throughout Canada, US, and Mexico. That's how we define North America, of non-lichenized fungi. What we ended up finding was, out of those records, there's about 44,488, not about, that's what we found, fungal taxa in these 3 different countries. Almost half of those are type specimens, which does give some legitimacy to the data, right? Because if it's a type specimen, you can't really argue about the identification. It's a type specimen. So almost 22,000 of these were actually tied to type specimens. And about 20,000 of those are macrofungi, okay? So slightly more microfungi than macrofungi. But those are just some highlights from that paper. We recently just published one a few months ago called the MyCoPortal. And if anyone's familiar with the R package and does statistics with R, you can now go on and use your R program to grab data out of the MyCoPortal. They talk back and forth, and you're able to grab that data and use it in R. So again, like I didn't want to recreate GenBank or MyCoBank, I don't want to recreate all the statistics that R already did for us. So what we do is just we're able to serve that data to R, let you guys that are experts using R analyze the data and as you see fit, and publish and cite the MyCoPortal, please. I appreciate that. All right. So just to wrap up my talk, I just wanted to mention some of the education and outreach. We're starting to get some buy-in from different mycology courses. This one at Michigan State and at Purdue University Northwest, where they contacted us and say, hey, you know, we do a mycology course. I have the students sequence 1 or 2 specimens, and they put it in GenBank. And yours is an ideal resource for doing that, because think about it, the students are learning collecting techniques, they're learning natural history, they're learning about herbarium accessioning, they're learning about Darwin core fields, now they're learning about sequencing through Sanger sequencing, they're learning about submitting that sequences and source modifiers through GenBank, and how to link all of this data together. So there's a lot of benefits to using the MyCoPortal in your classes. Another thing is the North American Mycoflora project that's just kind of getting kicked off. There's about 100 different mushroom clubs around the US. This is kind of one shining example where someone's put the data in the proper places and linked it up. So this is a public search interface. This is your metadata I showed you. But a lot of people like to use, especially the amateurs, or citizen scientists like to use Mushroom Observer, which is fine. But here's the specimen residing at the University of Arizona. If you click on genetic data tab here, you're going to get this with a URL to GenBank that I just showed you. So here's GenBank. Here's your link back to the Mycology Collections portal, which will link back to this page right here. If you click on Mushroom Observer, that's a hot link right there that hot links to Mushroom Observer. And from there, where the citizen scientists put up a lot of their images, that's where you can grab a lot of these different images of the morel that this thing has been sequenced. It's been, a specimen voucher is actually deposited. All the data, or most of the specimen metadata, is in GenBank, and you can go to, you know, a citizen science social media thing like Mushroom Observer or iNaturalist and look at and get more information. But at least it's all tied and linked together. There's also a link here on Mushroom Observer on MyCoPortal, which will link you back to here. So you got a link back here, you got a link back there from GenBank. So it's kind of a nifty little way to tie all of this data together. And then last but not least, I'd be remiss if I didn't say that we actually save lives with the MyCoPortal, right? Thanks to Kathy Hodge, one of your local own right here. So there's a little kind of story here that we wrote up. They had contacted Kathy about someone that ate a mushroom, and they thought, oh no, it's not the green gill lepiota. It's not this thing that will just make you really sick. Or they thought it was this thing that would make you really sick, sorry. She wasn't too sure, so she contacted Tim Baroni over at Cortland, and they worked together on this. And Tim went on and searched the MyCoPortal for Chlorophylla molybdites, the thing that doesn't make you sick in green here, versus Amanita virosa, which will make you sick and will actually kill you. And thank God they had Kathy and Tim working on this, because the doctors were going to send this patient home and just not even treat him. You'll get over it in a couple days. Well, with more data and research, they find out there's no green dots in New York. Chlorophyllum doesn't occur there. Only Amanita does. And that's what it turned out to be, and they ended up saving the person's life. So thank you very much, Kathy. I hope you got something for that. You did now. And with that, I'd like to thank you very much for your attendance. I know you all have a meeting in 5 minutes, I think, so.
Yeah, so if you need to leave for the meeting, you can, but if you want to stay and ask some questions, you should do that too. But can I just say something?
Sure.
So Andy's talking about the importance of these herbarium specimens. We have a big herbarium. We'll do half this process for you. So if you're working on fungi or any other plant pathogen, we would like your voucher specimens, and we'll help you get them into Andy's amazing network. Now, do you have a question?
Yes, sir.
Do you have any idea whether plant parents acidic fungi, because the earth is warming, as you mentioned, are spreading to new species as a parasite? As a parasite.
I don't have any knowledge on that right now. No, I haven't really looked at that data. I'm always interested in the next research question and trying to think those up. Sometimes that's hard to do, and then get the data out of the MyCoPortal. But no, unfortunately, I don't really have any expertise in that area, unfortunately. Yeah. Yes.
Can you say a little more about why undergrads are better than
the technology, better than PIs at sitting down and keystroking, or yeah, just in general. I mean, you do have a range of undergrad students. You know, some would do just 10 an hour, and some would do 40 or 50 an hour. But once you kind of weed out those ones that really are plugging and chugging away, they're really good at multitasking, it seems, compared to me. I mean, they can watch a video. A lot of them were watching a video, listening to rock and roll, and then keystroking in all at the same time. And by data checking, they weren't making very many mistakes by doing all of this. So I don't know. I'm not sure exactly why. I think they just seem to be a little bit better at multitasking and being able to focus on these things. And you know, they're growing up with keyboards, right? So they don't really know how to write in cursive anymore. They don't do that much anymore, or type on a typewriter. They know how to type on these keyboards, and they're just much better and faster at it. So we were very impressed with how much data we were able to get in from a large workforce of undergrad students. You know, hammering the MyCoPortal all at the same time. When you got 100 people on there, you know, you want to have a good network too to support all that. But luckily, the servers held up at Florida. Yeah.
Did you entice any undergrads to mycology?
Some, yeah. I didn't talk about that. There were some success stories where people were very interested in what they did, and I didn't know a herbarium existed. You know, I didn't know we had all these specimens, and they became more interested. We had a couple that actually went on and got degrees in collection management, and actually wanted to go on and be collection managers. So we're trying to train the next generation of collection managers, and we had a few of them buy into that, and they started out as digitizers, and now they're full-blown collection managers at different herbaria around the U.S. So yeah, we do have a few success stories like that, which is great.
All right, well, join me in thanking Andy.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.